Today we're going to look at viruses and prokaryotes. Let's take a quick look at classification. There is a huge amount of scientific data concerned with living things and their properties and characteristics. And the facts on their own as a data pile would be almost insurmountable, almost impossible to use. So to rectify, to organize the situation of this massive amount of data, we have split pieces of data or information into parts or sparsed it out into tiny packets. This organized system that we have built in order to sparse or organize this data pile is called taxonomy. Taxonomy is the name of this organized system. It's the science of dealing with the description, identification, naming, the classifying of organisms, and it's always evolving. It's not static. As more information is found or comes about, we will change or update the taxonomy. Let's look at some ideas of biological classification. Some classification systems are simple, but not all of them are. Here's some simple classification systems you should be familiar with. Organisms can be split into producers, consumers, and decomposers. Consumers can be split into herbivores, carnivores, and omnivores. And cells can be either prokaryotic or eukaryotic, and we'll talk more about each of those. Some classification systems are super specific, not simple. They're so specific that just by knowing the scientific name of an organism, scientists could be sure of its shared characteristics with others of the same species. Common names of organisms were used until the 1700s to discuss living things, but that could be a problem. Using the common name of organisms for a certain area, something that might be considered colloquial, like here in the South, we might say roly poly for a pill bug, that can be very confusing if we say are in a different area that doesn't use that terminology. It can make a classification system that's supposed to be even simple more complicated and hard to understand. So, for that reason, binomial nomenclature was developed. In 1735, Carolus Linnaeus developed a system of naming organisms that we still use today. He used the Latin based categories, genus and species, and because his organizational system included a two-part name, that's why we call it binomial nomenclature, a two-name organizational system. If you're using binomial nomenclature, you want to take the genus and the species name and italicize it. The genus and species name for each organism is specific. The genus is always written before the species, and organisms with the same genus are going to share some characteristics with each other. If they have the same genus and species, they're going to share even more characteristics. The genus name is also always capitalized, while the species name is always lowercase. We want to write the genus before the species. We want to italicize it. And we want to capitalize genus but never species. So, with that information in mind, which of the following is written correctly? A. Ursus arctos. B. Also Ursus arctos. Or C. Ursus arctos. So they're all the same genus species. We're not looking at that. We're looking instead for something that's italicized, that has the first word capitalized, and the second word lowercase. So if you said C, you would be correct. Next, let's talk about the 7 taxonomic levels. In addition to genus and species, Carolus Linnaeus categorized organisms in a total of 7 taxons. So he categorized organisms into these 7 classification systems, and we're going to call these levels taxons because of the taxonomy system he was utilizing. In Linnaeus' system, every species belongs to a genus. Every genus belongs to a family, every family to an order, every order belongs to a class, and every class belongs to a phylum. And then finally, every phylum belongs to a kingdom. So as you go down the ranks, each category, in the way that this is written, would become more general. But it is also generally written in the reverse, which would be more specific. So here we can see on the right-hand side, looking at the image that has the dog up top, we start at the bottom and we have domain, kingdom, phylum, class, order, family, genus, species. For the image on the right, below the picture of the dog, we are moving from the bottom up in order to become less to more specific. Oftentimes, the image or the classification system is reversed, and you'll start at the top and move down, which is the case for the images on the left in the table that is below the wolf and polar bear picture. Looking at the blue or shaded in blue parts of the graph on the left, you can see starting at the top and moving down, you have kingdom, phylum, class, order, family, genus, specific name, or species. So in the graph on the left in blue, we are moving from the top down and being more broad to more specific. A way to remember the order of these taxons from domain to species is the phrase, "Dear King Philip came over from Great Spain," with the D for domain, K for kingdom, P for phylum, C for class, O for order, F for family, G for genus, and S for species. So next we can actually look at one of these taxons, domains. There are 3 domains, and all organisms belong to one of the 3 domains, either bacteria, archaea, or eukarya. And a domain is just going to be the most inclusive or the broadest taxonomic category that there can be. It's even more broad than kingdom. A single domain can contain one or more kingdoms. For example, the kingdoms of Animalia, Plantae, Protista, fungi, those would all be within the domain Eukarya. Domain Eukaryota, or Eukarya, contains all of the eukaryotic cells, and eukaryotic cells have a true nucleus. Domain Bacteria, or Eubacteria, contains prokaryotes, and the oldest prokaryotes that do not have a nucleus, like all prokaryotes do not, would be in domain Archaea. So again, domain Archaea and bacteria both contain prokaryotes. Archaea is very primitive. These are older bacteria that can live in extreme environments. A lot of them are called extremophiles because they can live in really hot, really cold, too salty, or even environments without oxygen. These extreme environments that other organisms cannot live in. They also have cell walls that lack a protein called peptidoglycan, and therefore are not affected by antibiotics. We could also call the primitive archaeprokaryotes gram-negative bacteria or gram-negative prokaryotes, whereas bacteria, as a domain, has more advanced prokaryotes, more advanced forms of bacteria. Not all bacteria are pathogenic or cause disease, but the bacteria domain contains prokaryotes that have cell walls, and these cell walls specifically contain peptidoglycan, which makes their cell wall thicker. Peptidoglycan is a protein found in gram-positive bacteria, and peptidoglycan would be a component of the cell wall that allows these bacteria to be affected by antibiotics. An antibiotic is a chemical secreted by a living organism that kills or reduces the reproductive rate of other organisms. And of course, last but not least, eukaryota, or that domain eukarya, is where all life forms with eukaryotic cells would be found within. So again, still talking about the 3 domains of life, we're going to place organisms into the domains based on their cell type, prokaryote or eukaryote, their ability to make food, autotrophic, heterotrophic, and the number of cells in their bodies, unicellular or multicellular. So this information is what was used to put the kingdoms that contain organisms into the separate domains. Again, domain Archaea and Eubacteria contain prokaryotes, whereas all the eukaryotic organisms, including all of the multicellular organisms, are going to be in domain Eukarya. So organisms can be, again, prokaryotic or eukaryotic. Prokaryotic cells lack a nucleus. Eukaryotic cells contain a nucleus. Organisms can be unicellular or multicellular. A unicellular organism is single-celled, made up of one cell. A multicellular organism is made up of many cells or multiple. An organism can be autotrophic or heterotrophic. An autotrophic organism can make their own food, generally using sunlight or chemicals. A heterotrophic organism cannot make their own food and generally must consume or take in the nutrients they need. Looking at domain Archaea, we can see some prokaryotic organisms here. Notice that these prokaryotic organisms are all separate unicellular organisms. Domain Archaea has within it kingdom Archaebacteria that contains unicellular prokaryotic cells. Some are autotrophic, but others are heterotrophic, so we can't make an all or nothing statement about their nutrition processes. We do call these ancient bacteria. They tend to be older, but actually more similar to eukarya than they are to eubacteria. They are generally extremophiles, living in those extreme environments on Earth, things like thermal vents, hot springs, salty water, swamps, and the intestines of cows, where there's a lot of methane. Domain Eubacteria contains kingdom Eubacteria, and these types of prokaryotes are found everywhere, except in those extreme environments where you find Archaea. So they're not extremophiles. They do contain peptidoglycan in their cell wall. They tend to stain purple under a microscope, but they're unicellular prokaryotic, but they can also be autotrophic and heterotrophic, but always unicellular. And then we have domain Eukarya, which has many kingdoms: Protista, fungi, plants, animals, which are kingdoms Plantae and Animalia. And domain Eukarya consists of all the cells that are eukaryotic. This is the domain that has not only unicellular cells, but also multicellular organisms. For example, let's talk about the 6 kingdoms of life in general, and in order to do that, we have to continue looking in more detail now at the ones that are inside domain Eukarya. Let's start with kingdom Protista, or the protists. They're the odds and ends kingdom. Pretty much any organism that's eukaryotic that didn't quite fit in fungi, Plantae, or Animalia ended up in kingdom Protista. It includes any organism that cannot be classified as an animal, plant, or fungus. All eukaryotic. Most are unicellular, but there are multicellular protists. Some are autotrophs, others heterotrophs. So we can't make an all or nothing statement about their nutrition processes. And some have a cell wall, but it's not universal. Here are some examples of protists: the Euglena, amoeba, Volvox, and Paramecium. Looking at these cells, you can notice one thing: all of these protists are unicellular. Protists, however, are not always unicellular. Next, let's look at kingdom fungi. All these cells are eukaryotic, makes sense, they're in domain Eukarya. Most are multicellular, but an example like yeast is a fungus and it's unicellular. They are mainly decomposers. They break down dead or decaying matter, making them heterotrophic, or something that takes in nutrients for energy, including yeast, molds, mildews, mushrooms. Those would just all be some examples of fungi. And their cell wall does not have peptidoglycan. Instead, it has chitin, which is a thick polysaccharide of cellulose. Plants are going to be the next kingdom we look at. All plants are multicellular, eukaryotic, autotrophic, meaning they use sunlight to generate the nutrients they need for energy. A lot of them are terrestrial or live on land, and their cell wall is pure cellulose. Animals are also multicellular, eukaryotic, heterotrophic, and they live in many diverse environments, including terrestrial and aquatic habitats. So having looked or done an overview of those taxons, the 3 domains, and then looked at the kingdoms, next we're going to move on and talk about biological keys. Biological keys can help you classify organisms without having to memorize the characteristics of all the groups within the classification scheme. Biological keys are a series of questions that you can answer by looking at the major features of an organism you are studying. And then based on that answer from the organism you're looking at that you are studying, you can figure out what you're looking at, essentially, you'll eventually run out of questions. So, at that point, you should have classified the organism as well as the biological key would allow. Some biological keys are more specific than others. They're also known as dichotomous keys, but regardless, the keys are generally written as a set of choices, each involving two statements, and that would lead to the name of an organism. Scientists would use these to identify unknown organisms. For example, question 1, does your organism have dry skin? If no, then you're most likely looking at a salamander here. Does your organism have dry skin? Yes. Does it have hair? Also yes. Opposable thumbs? Yes. It's bipedal? Yes. If you answer yes in that order, that would lead you then to the human. Let's try one more. You have an organism that has dry skin, an organism with opposable thumbs, but an organism who is not bipedal. That's going to be, do it one more time. Has dry skin, has hair, has opposable thumbs, but is not bipedal. So your organism there should be the gorilla. There's other dichotomous keys that may look something like this, where you're given organisms and they're given general names like bird W, X, Y, and Z. And you're trying to figure out what is their species name? What is their more specific name? So you'd answer questions in twos to best figure out what organism is which. Let's see if we can figure out which organism is bird W. The beak is relatively long and slender, or the beak is relatively stout and heavy. Well, the beak is relatively long and slender, so it's most likely Certhidia. But what if it was stout and heavy? Then we would look at the bottom surface of the beak, and then that may lead us to question set 3, where we look at the lower edge. So let's try to figure out bird X. The beak is relatively long and slender, or the beak is relatively stout and heavy. I would have to go with B. That says go to 2. All right? On 2, it says the bottom surface of the lower beak is flat and straight, or the bottom surface of the lower beak is curved. For bird X, definitely curved. So if it's curved, it says go to 3. Let me go to the question set for 3. For 3, the lower edge of the upper beak has a distinct bend. And then for 3B, the lower edge of the upper beak is mostly flat. Mostly flat. So that would mean that bird X is the Platyspiza. Let's continue on and talk more about archaea and bacteria, which are both going to consist of prokaryotes, the tiniest living structures, and they actually make up the largest number of microorganisms. So even though prokaryotes are small, especially compared to eukaryotic cells, doesn't mean that there's not a lot of them. Their biomass is over 10 times that of eukaryotic cells. All the organisms in domain archaea and bacteria are prokaryotes, whereas we just have one domain with eukaryotes. So it should make sense there's actually more microorganisms or prokaryotic cells than there are eukaryotic cells. So looking again at domain archaea, it's difficult to study. They live in extreme conditions. It's hard to study the methanogens, halophiles, and thermophiles. Methanogens live in places with oxygen-free environments. They might live in the digestive tract of cows. They could live in garbage dumps. A byproduct of their metabolism is methane, which creates their environment that becomes incredibly oxygen-free and anaerobic. Halophiles like to live in extremely rich salt concentrations, such as Utah's Great Salt Lake. And thermophiles like really hot or really cold areas. For example, some archaea can live in the hot springs of Yellowstone National Park. There are both beneficial and harmful bacteria, so whenever you hear the word bacteria, you probably think immediately something that causes disease, something harmful or not good. But in domain bacteria, they're just prokaryotes that aren't quite as old as the ones in domain archaea. And the term pathogen would be used for any bacterium that is harmful. It's something that causes disease. But again, not all bacteria are harmful. So let's do a basic bacteria cell structure review. This is basically a review of prokaryotes. Because bacteria are prokaryotes, which means they have no nucleus and no membrane-bound organelles. They do have some ribosomes, and they do have a nucleoid region, but not a true nucleus. There are some capsules on certain bacteria. Capsules would be a covering over their cell wall that are generally sticky. They could help with adherence to surfaces, but also could be protective. Protective in the sense that pathogenic bacteria could protect themselves against immune cells. And the composition of their cell wall in domain bacteria generally includes peptidoglycan, but depending on the type of prokaryote we're looking at, we can use the presence or absence of things like the peptidoglycan protein to classify distinct types. For example, cell membranes always underlie the cell wall, and they're made up of phospholipids and proteins. We're going to see ribosomes in that cytoplasm. The central region again has the DNA, which is the nucleoid region, but not all have the same exact composition too. Their overlying structure for the cell wall, which is protective and generally contains peptidoglycan. In addition to a single circular chromosome, bacteria does also sometimes contain plasmids. Plasmids are additional pieces of circular DNA that can sometimes grant a bacterium some extra characteristics that help it survive in stressful situations. You might see fimbriae, long hair-like structures also used for adherence or grasping on some prokaryotes. Longer fibers, called pili, are sometimes part of bacterial reproduction. And for movement, generally there is a flagella, or flagella are present on a lot of prokaryotes. Again, pili are protein structures that extend from the cell that help bacteria adhere to surfaces. Flagella produce a rotary motion, basal body, hook, and filament are its parts. Here's an image of some of that. The bacterial flagellum here, you can see that it goes through the plasma membrane and the peptidoglycan layer of the cell wall, and even through its outer membrane, and protein rings are embedded throughout these membranes, and then you have the hook and filament structure create the entire flagellum. In order to identify bacteria, we can look at a couple of classification systems. We can look at the three basic shapes for bacteria: Coccus, Bacillus, and Spirilla. Cocci are round-shaped or spherical. They can form long chains or clumps. Streptococci is the chains. Staphylococci is the clumps. Bacillus are rod-shaped bacteria that can pair up in twos or even larger chains. And Spirilla are spiral-shaped. A spirillum is a rigid short helix, whereas a spirochete is a much longer rod-shaped flexible helix. Again, if you add the prefix strepto, you're talking about bacteria in a long chain. If you add the prefix staphylo, you're talking about bacteria in a large cluster. That's because these prefixes are included to indicate the arrangement of the bacteria. And a lot of this has to do, again, with the shape of the bacteria. The information is in the name. You might also see prefixes like di, tri, tetra. They would also be used to describe smaller arrangements of bacteria or small clusters. Examples you may have seen: Streptobacilli, Diplococci, Staphylococci. Diplococci is two spherical bacteria attached together. Streptobacilli is rod-shaped bacteria that is appearing in a long chain. And Staphylococci is spherical bacteria that is appearing in a large cluster. So here is a Micrococcus. This is a spherical bacteria. Here is a Bacillus or rod-shaped Salmonella bacteria. And here are some Spirilla or Spiroplasma bacteria. So you can see the general shapes of bacteria that would classify themselves as either Coccus, Bacillus, or a Spirilla. Next, let's look at bacteria's cell wall structure. And while studying and staining bacteria in order to make them show up better in a microscope, Hans Christian Graham discovered that some bacteria looked purple bluish, while others stained or looked red pink. And that's when he viewed it under the microscope. That's why we call the staining of certain types of bacteria Graham staining, because of his observations. The cell walls in bacteria contain peptidoglycan, and that allows them to easily absorb violet dye. So think peptidoglycan purple or peptidoglycan positive, because it's the gram positive bacteria that, when dyed, appear purple. They appear blue purple after a gram stain, and that's because they contain peptidoglycan in their cell walls. Gram negative bacteria appear red pink because they do not contain peptidoglycan, or in some cases contain a very thin layer of peptidoglycan. With less peptidoglycan, there is going to be less of an absorption of the stain, meaning that their color is red pink, and it generally sticks just to the outer membrane. The capsule that you might find on some bacteria does surround some cell walls, but doesn't prevent the staining of the bacteria. So here's a gram-positive cell wall. It has a thick peptidoglycan layer, and then we have the plasma membrane under the cell wall with a thick peptidoglycan layer. Here's a gram-negative cell, and its cell wall has an outer membrane of lipoproteins and a thin peptidoglycan layer. It's actually protected by that outer membrane. So gram-negative bacteria would have a cell wall still that covers the plasma membrane, but gram-positive would be purple, gram-negative would be pink. So here we can compare both. Gram-positive purple, gram-negative pink, gram-positive peptidoglycan more present, less peptidoglycan or none for gram-negative. These are all going to be in domain eubacteria or bacteria. So because of that, they must contain some peptidoglycan, but we could say that those in domain archaea, which have none, would most likely also stain gram-negative. And that's where we're going to start, or stop, because we're going to start next here talking about producers, consumers, and decomposers of domain bacteria. We'll go into another classification system as opposed or different to the differences between prokaryotes and eukaryotes.